Biomarker discovery and validation is vital to the future, both of the pharmaceutical industry and biotechnology industry, as well as to us as patients, which we all are in the future. It's important from an early perspective as understanding our diseases better so that we can identify the targets that we're working on, all the way through to selecting the right patient populations as we go through the development program so that we have a much higher chance of success and seeing efficacy within the programs. And as we move towards an era when we are much more targeting the disease process itself, modifying the disease rather than trying to treat the symptoms, it's vitally important for us to have good, well-validated biomarkers, because these trials to modify the disease symptoms take a long time and are very expensive, and we need to build confidence early in the program to be able to invest the amount of money needed to see disease modification, which may take a couple of years or even longer to see. Personalised healthcare has many definitions in the literature. At AstraZeneca, we define personalised healthcare broadly as being able to use any tool, be it a new biomarker, be it standard clinical measurements used in a different way, to improve how we can select patients so that they get the best possible therapy for them. In terms of how close are we to achieving personalised healthcare, I think there are some real strides that have been made forwards. We can see within the oncology therapeutic area that we are now making advances at a much faster rate, being able to specifically target certain mechanisms that are the main causes of cancer in certain patients. However, in other areas, we're still a little bit further away. The science needs to develop, but we are getting the infrastructure right, and we are seeing new breakthroughs day by day. So personalised healthcare is going to be vital to our future, and in order for us to be successful and to create personalised medicines, we do need high quality samples that are well validated and provide us with the highest possible biological information so that we can select the right patients going into the trials, and as we move further forwards we can use those samples with the clinical information attached to it to develop the best possible diagnostic tests. Good biobanking is absolutely critical to effective biomarker research. We know that where we get high quality biological samples with good clinical information, the data that we generate can be validated and we get real biomarkers that stand up as we test them again and again. In the past, where we have not found biomarkers to stand up to multiple testings, one of the key challenges has actually been the quality of the biological material and the link between that material and the clinical data. So biobanks and the people responsible for them could improve our biomarker research by increasing the number of high quality samples we've got and the quality of the clinical data, both in terms of the amount of data we get for each of the patients and the samples they give, and also for collecting that data in a long-term manner so that we can see how the disease is progressing as those patients get treated and get older. So if I could change one aspect of this whole process around, as we said, the biobanking from the sample acquisition through to the clinical validation at this point, it would be really around suddenly having access to a much broader range of tissues from patients with a broader range of diseases at various stages and from various different ethnic backgrounds. Because where we are at present is that we still have limited numbers of samples from limited ethnic backgrounds and limited diseases, and that is holding us up from really driving the science forward and improving the quality of healthcare and the drugs for all. So what's stopping us from changing it is actually just the speed and the level of investment which is required to get the breadth of samples and clinical data we need. We're not talking here of tens of hundreds, we're talking of tens of thousands of samples that we need in each of the different disease areas. So we at AstraZeneca, like many other pharmaceutical companies, are investing in biobanks, are investing in sample collection and the collection of high quality clinical data, but it takes both time and money to build the size of biobanks that's required for us to make the steps that we need. So in my role as VP of Personalised Healthcare and Biomarkers, my top 3 priorities for the next 12 months are around building the evidence base and the molecular signatures of the key cancers that we are developing new therapies to at AstraZeneca, as well as developing those molecular signatures which will drive forward our development programs. We also have important programs in other areas, in neuroscience and also in inflammatory diseases and anti-infective diseases, where we are looking again to build the strong evidence base, the biomarkers that have got clinical validation and clinical usefulness, so that we can prosecute our key high priority development programs and get new therapies out to the right patients.